Welcome back, everyone. Hope you all joined through the link provided through WhatsApp. Link for the first lecture will be given through WhatsApp group. So please join through the link, corresponding links. So now it's time for the first lecture of the series. I call upon Ms. Fatima to introduce our first lecturer, Dr. Bhaskar Saha. He will be presenting the topic Basic in Brain Development, Adult Neurogenesis, and its Therapeutic Potential in Neurodegenerative Disease.
Fatima, please unmute yourself. You are not audible. Fatima, you are not audible. Fatima, I think it's mute. No, Fatima. No, no. Can't hear you.
Dr. Bhaskar Saha is Assistant Professor, Department of Life Sciences and Biochemistry at St. Xavier's College, Mumbai. He has completed his BSc from Presidency College, Calcutta, and from MSc Biotechnology in MS University, Baroda. He has done PhD in Life Sciences, Tata Institute of Fundamental Research, Mumbai. He has done his postdoctoral research from 2006 to 2010 in Institut de la Vision, Université Marie Curie, Paris, France. The project was a role of Axon guidance molecules in adult neurogenesis. From 2010 to 2013, the Experimental and Clinical Neuroscience Laboratory, University of Poitiers, France. The one project was on stem cell based therapy in Parkinson's disease. In addition, he worked in a project on deriving cortical neurons from embryonic stem cells and their use in the repair of damaged cortical neurons in mouse brain. He has done 11 reviewer for several journals. The honors and awards he had scored is Nehru Foundation Scholarship for Research Assistant Professor position at the Nagoya University, Japan, Postdoctoral Research Fellowship, and Conway Rickey Fellowship for Career Development from TFR 2002 to 2006. Over to you, sir.
Thank you, Ms. Madhuna. Am I
Yes.
audible to you all?
Yes, sir. Yes, sir.
Okay, okay.
Audible.
Is the slide visible?
Yes, sir. Slide is now visible.
Okay, okay. I'm just making it full screen. Okay. Okay, good morning, everybody. My name is Bhaskar Saha, and I'd like to thank the organizers for this conference, Dr. Susan Ma'am and Dr. Nilima, and the UC College. And I'd like to congratulate the UC College for their centenary year. And I'm sure they're going to organize several such conferences for the students and for other people around. Okay? So as I see that different lectures for this topic are mostly psychology based, so my talk will be a kind of a precursor to understand the basic nervous system, the functioning, so that the later talks becomes much more easier to understand in terms of its functions. Because when we hear the later talks, we have a background already that what the brain structure is like, what is the neuronal connections are like. So, and what happens in different neurodegenerative diseases, and that might lead to psychological defect. Okay. So with this prelude, what I try to do is first give you a basic flavor of the uh, neuronal development process in mammalian system, and mostly it will be on the rodents and the human being. Okay, so people who know about the general aspect of the development of an embryo or an embryogenesis process, we know that there are three germ layers called ectoderm, endoderm, and mesoderm. And these three germ layers give rise to the entire body plan. And these three germ layers form from a very small pool of cells called this inner cell mass, which is here. And then this inner cell mass, a particular group of cells of this inner cell mass called epiblast cells, which in fact divide and differentiate into different cell types and ultimately form the entire embryo. Okay. So the gastrulation process, I am sure some of you or most of you have heard this terminology, is a process which shows an extensive cellular migration along with cell division process. And this migration is necessary for making these three germ layers, ectoderm, endoderm, and mesoderm. Okay, so if you follow the arrows over here, this ectoderm, the cells, they move towards the middle part and then invaginate and move downwards. So here the blue color is the animal pole region where most the cell migration and cell division is happening. Yellow is the vegetable pole area. Then once the cell starts moving down, the upper greener part is going to form the future ectoderm. The yellow part is going to form the future endoderm. And in between, when the cells migrate inside, they just form a third layer between these two green and yellow layers. And this purple layer is called the mesoderm. Okay. And now, if you look at these structures, so now, as I said, that there are huge cell migration that starts at this point of time. And this cell migration leads to the formation of different structures. Initial structure that is formed in the middle is called a notochord structure. Now, I'm just trying to orient you in this 3D structure. So if you see, it's a cross section. So suppose you have taken a section of that embryo. On the other side of my computer is the front part of the embryo. Towards my side is the back part of the embryo. So anterior to posterior. Okay, so head to tail region. Okay? And if you have taken a cross section, then you see this ectoderm, mesoderm, and endoderm. Now, suppose I now hold these two ends, the two arrow which is shown here and here, and pull the entire thing down, all the way down, and fuse it at the bottom, what I get is a structure of this sort. Okay? And that is how the entire embryo is formed. When it formed, this outer green line or green area is the ectoderm, which is going to form the epidermis and the central nervous system. So when we say central nervous system, it is the brain as well as the spinal cord structure. Okay. And then this yellow part is the endoderm, which is the gut system. So this is the entire GI tract at the central whole structures. And this is the coelom or the body cavity, which forms the pleural cavity, that is our chest cavity, and the peritoneal cavity, that is the belly cavity. And rest of the structures form here. Okay, so this is just to orient you about how a developing embryo look like in a 3D situation. Now let's come back, or let's just go into the nervous system development aspect. As I told you that the ectoderm, it shows a severe or extensive movement process. And this movement is something called a convergent extension process of movement. And as it moves, it forms a tube-like structure. So if you just remember the previous picture, we had an ectoderm which is a planar structure, a sheet-like structure, okay? And now if you push this structure from both sides, it will fold. I'll just give a small example. So I have a piece of paper, okay? And if I now try to push the piece of paper and then, bring it close, both ends close together, what I see is a tube-like structure at the bottom and the upper planar structure, okay? So exactly the same thing happens here. And once this happens, you get a tube-like structure, which is formed, which is called the neural tube structure. So it gets converted from a neural plate to a neural tube structure by this folding process, okay? Now, if you go look at this black panel at the left-hand side, these are the different ages of a developing mouse embryo. Mouse embryogenesis takes place for around 18.5 days, okay? And the nervous system starts developing from day 8.5. So here, I'm just trying to orient you about this embryo. This is the head part, this is the eye, this is the front part of the brain, this is the middle part, and this the entire structure is the spinal cord. Okay, and now this brain and the spinal cord is a single structure. They are not two different entities. It's a single entity. At the front part, the entity is called the brain. At the rear part, it is the long tube-like spinal cord structure. Okay, so and just to tell you, I told you earlier that when we're talking about the ectoderm, it gives rise to the central nervous system. But then we have nerves in our, say, limbs, forelimbs, legs, okay, all around the body. So where from these neurons come? These neurons constitute the peripheral nervous system. And this peripheral nervous system comes from a very highly migrant population of cells, which are called the neural crest cells. Now, if you follow this structure again, we have this blue entity, which is going to form the future epidermis, that is skin in our case, or maybe feather in case of birds, or scale in case of fish, okay? And then there's a small patch of these green colored cells, and then there is this purple cells, okay? So these purple cells are going to form this neural tube structure. These green cells, they fuse with each other, and then they just migrate. And these are the cells in a developmental biology term, they are called the mesenchymal cells. And these mesenchymal cells are very, very migratory population of cells. And they form diverse cell types later on. Okay, they form the enterochromaffin cells, they form the interperipheral nervous systems, etc. Now, how is this neural tube formed? How this neural tube closes? Because this is one of the interesting aspect of the nervous system development. See that when we are talking about developmental biology, all the different systems, our heart develops, everything develops, but nervous system has some unique properties, and that sets it apart from the other developing systems. Okay, so my aim today will be to talk about those unique properties of the nervous system development, which are not generally seen in other organ development process. Okay, so I talked about that a tube is going to form, and at the front part of that tube, the brain will be formed, and the rear part, the spinal cord. Now, if you think of a bag, okay, and you need to close this bag, or you need to close a jacket, either you can have a zip, which you start closing from one end and then gradually come to the other end, okay, or you can have buttons where these buttons are intermittently placed. Okay, and suppose you want to stitch up the entire thing, first to put on the buttons, and then you stitch the intervening regions, and that's how you can close the entire shirt. As opposed to if you have a zip, you can just pull the zip from one end to the other. So the idea was, or the developmental biologists were thinking, what way this neural tube closes? Either it's a zipping mechanism or a kind of a button mechanism. So initially it was thought that it is a zipping mechanism, and the development and this closure starts from the very front part, that is from the brain, and then goes gradually to the spinal cord, at the end of the spinal cord, near our hip region. But now we know that it is not correct. The first closure of this neural tube happens between the junction of the midbrain and the hindbrain region. I'm sure most of you know that the brain has a forebrain structure, then a midbrain structure, and the hindbrain structure. So it happens between the midbrain and hindbrain, and there are some intermittent areas where the initial closure takes place, and then rest of the areas, they get sutured up. Okay, and that's how it got closed. So this is a process of neural tube closure, which is something called a primary neurulation, because this is the most important or the basic mechanism of how the neural tube closes. Because when the neural tube does not close, there are several defects, birth defect that takes place, and in most of the cases, the babies are perinatally, they die perinatally just after a few moments of birth, or maybe a few days to few months of birth, or even the prenatally, embryonically, they also die. So the neural tube closure is a very, very important aspect. So the primary neurulation, it suggests that it forms a hollow tube-like structure. But when it was seen that all the entire vertebrate systems were scanned for the neural tube formation, it was seen that it's not always the case. For example, in case of fish, it is completely different. Even in mammals, there are some other mechanisms as well. And what is that mechanism? That is something called a secondary neurulation process. And what is this secondary neurulation process? Here, it starts from a solid tube. And how this solid tube is made? The solid tube is made by these migratory mesenchymal cells. Okay? They condense and form a solid tube. And then this solid tube, the cells at the center, they undergo apoptosis. Okay, so these cells at the center, they start dying. And as the cells start dying, you get a central hollow structure. Okay, so you start with a solid tube, and then gradually you just, the central cells die and you get a hollow tube. Similarly, what happens during a bone development? For example, the bone in the arm, which is called a humerus bone, is also same way their bones are formed. They are hollow bones, right? And how this hollowness is formed? Simply because initially there's a solid bone, then the central part does get apoptosis. So it's a similar mechanism. So there are different mechanisms of this neural tube formation, and how this closure happens, at what position this closure will happen is not a random process. It is governed by some adhesion molecules. Okay, and these adhesion molecules are calcium dependent, or is called cadherins. So it has been seen that the ectoderm, which is going to form the future epidermis, skin structures, they are synthesizing E-cadherin molecules, whereas the future neural tube area, they're expressing N-cadherin tube. And we know now that the cadherin interaction is a kind of something called a homophilic interaction. That is, the similar molecules attract or bound to each other more strongly than two different types of molecules. So there are experiments done, and this is just a cartoon of that experiment. So if you have a mixture of cells, you have individual cell suspensions, and you just have these cells, both E-cadherin and N-cadherin type. And if you keep these cells in the culture system for a few days, you will see that all the E-cadherin cells, they cluster together, all the N-cadherin cells, they cluster together. So this is something called a homophilic interaction, and that is very, very important for this neural tube formation structure. Okay. Now, as I said, that if there are a defect, and that defect can be very drastic, there are certain molecules. I'm not going into the molecule, like some proteins called proto-oncoproteins. These proteins are necessary for the embryonic development. And why they are called proto-oncoprotein? Because if there is some mistake happens in these protein structures, their function becomes abnormal. Now they act as a oncoprotein, okay, or leading to cancer. And there are some other molecules which are very, very important in the early development. And if these molecules are defective, then the brain is not formed. So here, this is a condition in this left panel B, which is called exencephaly. That means neural tube closure does not happen, so the brain is an open brain condition. In case of this SHH mutation, I'll come to that molecule later on. This is something called a holoprosencephaly. That means we know that our brains are of two different hemispheres. Here, the two hemispheres do not form. They form a single hemisphere, but as this molecule is necessary for cell division, so the entire size of the brain is very, very diminished, almost negligible. Okay, so these are very drastic differences. I'm just giving you two such examples. There are several such examples which shows developmental defect, and because of this this defect, these embryos, they do not either they do not born alive, or if they born alive, they die immediately after. Now I'm going to tell you a basic mechanism how this entire development happens. For example, when we talk about the ectoderm, it is all the cells initially the same type of cells, right? And then after some time, some cells behave differently from other cell types. And if you just try to imagine that why our heart is different from our liver cells or different from muscle cells or so on and so forth, because all of them have started from a single pool of cells which all were identical. Okay, so there has to be some kind of a molecular hierarchy, okay, or a very stringent molecular function which leads to this kind of a different fate of these different cells. One of the very early group of molecules are called morphogens. What are these morphogens? They are diffusible molecules. To give an example, suppose you have a bottle of perfume, and if you spray the perfume from this point, the person who is standing next to you will get the highest concentration of perfume. And if the perfume contains alcohol, that person will get a more alcoholic smell. But the person who is standing very far will get maybe a floral smell. It's the same perfume, but as it has diffused over a length, so different people standing at different lengths, they are perceiving different properties of this perfume. So morphogens act like this. So here, if you have these different blocks as a different pool of cells, and this is a morphogen source, okay? So then the cell which is close by will get a different effect of this morphogen, of this molecule. The cell which is at the furthest, they get a different effect of this morphogen. And how is this different effect takes place? It takes place because when this molecule bind to the receptors on the cells, they activate certain group of proteins. And what are these proteins? They are called transcription factor. And I'm sure most of you have heard of this term called transcription, which leads to gene expression, okay, and ultimately forms different proteins. So the cells, they're getting the same morphogen, maybe in a different concentration, but ultimately these cells are now starting to express different sets of protein, and that will lead to the different fate of these cells. So this is a general outline how the developmental system functions, okay? And to give you one example, in this neural tube closure, we have two morphogens. I'll come to these names. The BMP stands for bone morphogenetic protein. SHH stands for sonic hedgehog. Okay. Sorry. So these two molecules, they act in an opposite concentration gradient. And because of this opposite concentration gradient, at certain point, they lead to this hinge formation. Because this hinge is necessary for the closure to happen. While the BMP, the upper molecule, helps in the formation of the hinge, and the lower molecule, sonic hedgehog, actually prevents the hinge formation. Okay, and now when the cells at this hinge region get a proper concentration of both these molecules at the optimum concentrations, the hinge is formed at this space, okay, and leading to neural tube closure. Now, next thing that is very interesting is how the brain, especially, has different segments. We have a forebrain, midbrain, hindbrain, okay? And their structures, their organization, their function, all are different. Why is it like that? So again, when we look at the developmental mechanism, there are these morphogen gradients that are very, very important. So this is a very classic picture of a embryo, mouse embryo of around day 14.5 or so. So when people studied through different assays, what are these morphogens that are present in the different parts of the brain, they found that morphogens like fibroblast growth factor, which is present at the very front part, then the sonic hedgehog, which is present at the lower part, some other molecules which is present at the top part. Okay, and because these molecules are present at different areas, they set up gradients, and that gradients lead to formation of this axis. What kind of axis? We have a body axis called, which is from the anterior, that is head to tail or toe in our case. So that is the anterior posterior axis. We have a dorsoventral axis where the back side is the dorsal side and the chest side is the ventral side. So these body axes are formed. Similarly, in the brain also, these axes are formed, and they're very, very important functionally. I'll just give you a couple of examples. Okay. Yeah. So just to give you one experiment, how people have understood this axis formation. This group, Elizabeth Grove's group, they studied how the anterior posterior axis is formed. Now, I'm sure all the biology students, including the psychology students, they know that motor neuron, you have a motor area in the brain, then the somatosensory area in the brain, and then the visual area of the brain. And so they have a certain segmentation, okay? Now, what they did is to study the role of this fibroblast growth factor, how they regulate this regionalization, why the motor neuron, motor cortex is here, somatosensory cortex is here, and the visual cortex is at the back. So what they did is they used some nitrocellulose beads, okay, the small beads which may be a few micron size, and then they soaked in this fibroblast growth factor solution, and then they put at the front where the asterisk is, okay, and then observe what happens to this regionalization. When they put these beads here, that is, they are giving an extra source of fibroblast growth factor. So the net level of fibroblast growth factor here is more than normal. So in that case, they found that the motor cortex area, which is at the front of the part of the brain, they expand. And the somatosensory cortex push back, and visual cortex is very tiny at the very back. So then they did an opposite experiment where they inhibited the effect of a fibroblast growth factor. And what did they see? They saw that the visual cortex now expanded, the somatosensory cortex moved much forward, and there is hardly any motor cortex. So what they suggested from this, or this experiment suggested, is that if we need a certain amount of this fibroblast growth factor for this region specificity in the cerebral cortex. And FGF is helping for the formation of the frontal part of the brain. So if we increase the FGF concentration, the frontal part becomes bigger. If we reduce the FGF concentration, the frontal part becomes much lesser, and the back part becomes much bigger. Okay, so this is how, and just to orient you, this is if you are looking from the top of a mouse brain structure. So this is how that, I'm just giving a very small example of how anterior posterior axis regulates the region specificity. Similarly, the dorsoventral axis, like upper part of the brain versus lower part of the brain, like cerebral cortex versus other areas of the brain. Okay, and this is very, very prominent and easy to study in the spinal cord, because spinal cord is a rod-like structure, right? So when people looked at the spinal cord, and if you remember our previous pictures, that we have a top part where the epidermis is, then the neural tube-like structure, and below I first talked about that another structure called notochord, okay, which is the early developing structure. Now, when people looked at this structure, what was found that the upper part or topmost area of this neural tube, they synthesize the molecule called bone morphogenetic protein 4, and the lower part synthesizes sonic hedgehog. So what will happen? They are diffusible molecule, so they create a counter concentration gradient. Sonic hedgehog is highest at the lowermost part, and BMP is highest at the topmost part. So the topmost part is called the roof plate, as if a roof of a room, and the lowermost part is called the floor plate, floor of a room. Okay, and now if we try to correlate with the how the spinal cord looks like, and this is a cartoon of a cross section of a spinal cord, we know that the sensory neurons are present at the dorsal end, motor neurons are present at this lower part, and the interneurons present at the central part. So the segregation of the sensory neuron, motor neuron is not a random process. This is made by this initial concentration gradient made by this group of morphogen proteins. And the cells which were all along this length of the spinal cord, they got differential concentrations of either BMP or sonic hedgehog, and based on this differential concentration, they form different cell types. Okay, so this is one very important aspect of brain or the central nervous system development. The next important aspect or fascinating aspect is how our cerebral cortex is formed. Cerebral cortex is a layered structure. Okay, what do you mean by layered structure? As if you have a one layer of cells which is stacked up on the other, then they're stacked up on the third, fourth, like that. So our cerebral cortex has 6 such layered structure. Now the question is, how are these layered structure formed? And remember one thing, the entire brain is not a layered structure. Okay? It's just some part is layered, some part is not layered. For example, I'm sure the psychology people know about the limbic system. Limbic system is a accumulation of different structures. Okay, say the structure called basal ganglia, the structure called caudate putamen, or nucleus. I'm just taking all these structures, or the thalamus structures, which are very prominent structures. They have the clusters of cells. Okay, and their organization is completely different from the cerebral cortex organization. Okay. Now, let us look at how this layer of the cerebral cortex is formed. Okay, so this is a mouse embryo, which is approximately day 14 or day 15 time, and you have taken a cross section like this. You are looking from the top of a mouse embryo. And if you take a cross section and then look the embryo from the front, you will see there's two hemispheres like this. And here, the central part is the hollow area called the ventricle area, okay, which is filled with the cerebrospinal fluid. Now, if we have the ventricle, and I'm just going to the lower cartoon here, this thin part above is going to form the cerebral cortex, and the lower part is going to form the other part of the brain. And remember, I'm talking about only the forebrain structure, front, or not the midbrain or the hindbrain. Okay. Now here, the cells which is lining this ventricle, okay, is called the ventricular zone. Here, ventricular zone. And we know now that all the dividing cells, or so-called the stem cells, they are present in these ventricular zone. They are not present anywhere else. So the entire lining of the ventricles, starting from this point to this point, coming to here, everywhere we have these dividing cells. Now in the cortex, what happens is these dividing cells, when they divide, the division can be of two types. You can have a symmetrical division or asymmetric division. Let's not talk about that. Once they divide, they then starts moving upward or outward. It's called a radial movement. And how is this guided? This is guided by certain cells, certain glial cell types called the radial glial cells, which connect both the innermost area of the brain, of the cortex, and the outermost of the part of the cortex. To give you an example, suppose there's a 3-storied building and you have a pipeline and somebody wants to crawl up using the pipeline. You can imagine that person is crawling up. So here, the cells also crawl up against this railroad kind of structures made by these radial glial cells, and at one point of time they get detached. So suppose that person wants to go to the second floor, and it comes up to the second floor, finds the balcony of the second floor, and come out of the, or release, or just jump into the corridor, or sorry, in the veranda. So similar thing happens. Once they get to a certain level, they just get detached from this railroad of these glial cells and then settle down. And then the next wave of cells again come, and then they penetrate through the first layer and then go top and then again settle down. Okay? So this is how the cortical layer is formed. And when people looked at, is there any difference between the mouse or other vertebrates or a human being, the mechanism is pretty much the same. Okay, here we have the two cartoons here. Now, it is one kind of movement. There's another kind of movement which is called a tangential movement. That means the cells which are forming here, non-cortical area, they are synthesized here and then they move all the way to the cerebral cortex and they form the inhibitory interneuron. So I'm sure the psychology people or the other neural people who have studied neurobiology, then you know that we have certain neurons called GABAergic neuron, which are inhibitory in nature. So these GABAergic interneurons are synthesized here, and then they go all the way to the cerebral cortex. Okay, so this is how the cerebral cortex is produced. Now, the next, and I think the most important aspect of the nervous system is how an axon, okay, or how a neuronal process finds its target. Okay, and we know that there are several billions of cells within our brain, and there's a lots and lots of connections. But across individuals, these connections are very similar, very stereotyped connections. So how these connections are made, how a connection which is just starting to form, an axon just starting to form, knows where to go, which is the target cell. Okay? And that is very, very fascinating. So this person, Dr. Roger Sperry, who did a lot of work, and one of his work is to find out this kind of axon guidance process. He used frog, okay, So what he did is he used the frog, and then he cut the retina and then put the retina back by rotating 180 degrees. That means the front part goes down, lower part goes up. And in frog, the regeneration process is very, very efficient. So they again form back the neuronal pathways. And when she studied how the retina connects to the next part of the brain, that is in some cases called tectum, some cases called superior colliculus or lateral geniculate nucleus, etc., etc. So how they are connected. So it was found that, so what was the experiment? Sorry, the experiment was said, normally if you have a prey, or if the frog sees a prey, it flaps its tongue to catch the prey. Now, once he rotated the eye ball for by 180 degree and then let it heal, and then presented a prey, the frog flapped its tongue completely 180 degree opposite. Okay, so it suggested that the connections at the lower part or the later part remains the same. Only it changes at the retinal area, but when these retinal neurons regenerate, they can find their proper target. Okay, and that's why for that manipulated frog, the entire visual field is topsy-turvy. Okay, so we know that when the axon grows, the axon actually finds out this pathway. How it finds out this pathway? By interacting with the nearby microenvironment. Okay, so if some area is favorable, the axon will turn towards this side. If some area is not favorable, then it will turn away from this side. So this moving part is called the growth cone area, which has a very dynamic structure. It can extend, contract, and by interacting with the local environment, it can find out which way to navigate. Now we know that there are several sets of molecules that are regulating this chemoattraction process versus the chemorepulsion process. Now, to understand this example of this retina, now we know that there are a group of molecules called ephrin, and their receptors called F receptors. They act via the chemorepulsion process. Okay, so some neurons which are present here, they can go to high ephrin region, some go to lower region based on the concentration of ephrin from which these neurons arise, and then the receptor area. Okay? So it is the chemorepulsion mechanism which is prevalent here, and there are several such superfamily of molecules that are now identified which regulate this process of navigation. Okay. Now, this navigation, or how a connection gets stabilized, is very, very important. Okay? At the very beginning, or just after childbirth, the connections are very random. Then you need to prune it, like you prune a sapling to let it grow properly. And so before pruning, suppose you have a set of neurons here, and then this set of neurons connecting to here. So all neurons, they just randomly connect to all this next level of neurons. But then based on which connections are sending more stronger signals, that is more stronger electrical signals, those signals or those connections remain stabilized, and rest of the connections, they get retracted. Okay. So for example, after pruning, this neuron connects to this neuron having a very high signal. And this pruning is necessary for stabilizing. And that's why in babies, there are lots of abnormal or very abrupt neurological features that have been seen, which gradually get stabilized over time. Okay, and that is talked by Donald Hebb, who said that when two neurons, they connect to each other, the structural and the functional stability depends on how they are sending functionally connected to each other. Okay, so in layman's term, it's called the neurons that fire together, wire together, and that is very important. So it's an activity dependent process, because This is very, very important for the initial phase of learning in our life, which is called experiential learning in the critical period, that is early few years. And I'm sure Dr. Vidita Vaidya is going to talk about this in the later section. So this is about the neural development that I want to talk. Let's talk about the adult neurogenesis. Okay. Initially, we also knew during our school days that neurons are not generated in the adult brain, etc., etc. Same was told by Santiago Ramon y Cajal. But now we know that it is not true. Because if there is no adult neurogenesis, that is one of the very big roadblocks to any kind of amelioration of neurodegenerative diseases. Okay? So in 1965, Joseph Altman and Gopal Das first said that they looked at the hippocampus, the structure which is responsible for our memory process. They studied those structures and they found that there is some amount of neurogenesis, new neuron synthesis happens in the adult. Okay. And that was a very path-breaking experiment because that opened up a lots of opportunities for the neuroscientists. But then there was a lull for several years before, again, that aspect of neural research kicked up in a big way. First set of studies were done in songbirds. And why songbirds? Because it was observed that there are several species of songbirds like canary, like zebra finch, etc. During the reproductive time, that is in the spring season, the male birds, they try to attract the female counterpart by uttering or different types of songs. Okay, and it was observed that the singing pattern or the song pattern, the notes, okay, or the even they differ from one season to another. Also, the entire song pattern can be completely different, not only few notes, but entire song pattern. The intensity of singing, the accuracy of the song pattern also gets changed. And it's a cyclical process. It is very, very prominent during the spring, but later on it gradually goes down when the day length becomes lesser and lesser. So people try to understand why does that happen. So then Alvarez-Buela's group in UCLA, they studied this pattern, and they found that during the breeding season, there is a very high level of neurogenesis that happens in the particular area of the these male birds, which are called the high vocal area, the area which is necessary or which is responsible for the singing pattern. And this high vocal area connects to a certain area other called a robust nucleus of archipelium. Forget about the names. So this HVC area, RA area, very, very important. And there's a huge amount of new neuron synthesis that happens in this area. So the turnover rate is very high. Okay, so that was a very, very interesting study which suggested that yes, adult neurogenesis takes place. Later on, when they found that why it is only specifically in the male and not in female, female happens in very lesser extent, simply because it is guided by testosterone. Okay, with during springtime, with the increased day length, the level of testosterone goes up. And that activates certain growth factors like brain-derived growth factors, like insulin growth factor 1, etc., which are necessary for the new neuron synthesis. Okay, so if you have a high amount of testosterone, this neuron will synthesize. But during other time, when the day length is low and during other seasons, okay, this testosterone level goes down, which leads to the reduction in the or cell death in these high vocal centers. Okay, we know that now it's not completely true because testosterone gets easily degraded. So birds, this high vocal region has enzymes called aromatase, which converts testosterone to estradiol, and that is also necessary for this new neuron synthesis. Now, if you look at across different species in the vertebrate, the neurogenic region in the adult is different. The bird has a very high neurogenic region. The fish, they have several small neurogenic region. But in the mammalian system, they are very restricted, which suggests that as the circuit complexity increases, the chances of new neuron synthesis gradually goes down. In case of mammalian system, what happens? Let's try to understand that. So this is a cross-section of a brain. So we have a 3D cartoon. So if this is the mouse brain, we're looking from the side, and we take a cross-section somewhere here and look from the front, we see both the hemispheres. And here, if you remember, we talked about the ventricle. So this blue structure is the ventricle. And the cells, which initially we talked about ventricular zone, in the developing stage, now we are talking about saying the same region as a subventricular zone. Okay, the property is a little bit different. So this subventricular zone, when people studied, they found that again, it's the Alvarez-Buelas group. Primarily, the lady who looked into it is called Fiona Deutsch. Okay, they studied this property. They found that in this area where new neurogenesis happens, to give you a number, in case of mice, it is around 10,000 neurons per day is synthesized. However, for human being, the number is pretty less compared to the rodent system. And here, there are 3 types of cells. If you look at the periphery, you have these blue color cells, which are nothing but astrocytes, the glial cells. They are not neurons. And these astrocytes are the stem cells in the adult brain. These astrocytes give rise to very transiently proliferating cells called C cells. They are small cells, but they divide very rapidly. And then these C cells gets converted to A cells, which are the neuronal precursor cells or the neuroblast cells. And now these cells, which are generated here, okay, in this picture, they take a very defined route, which is called the rostral migratory stream because they're moving forward, and go to the olfactory bulb, okay? And in the olfactory bulb, they form interneurons. So they are not forming a part of the signal or the signal transmission pathway. Signals transmission pathway is fixed. What they are forming is a small interneurons in the olfactory bulb to modulate the signal strength or the nature of signal of these olfactory sensations that will be perceived by the mouse. In the mouse, the olfactory sensation is very, very robust, okay, as compared to us. So in human being, we have this system, but it is not as robust as the mouse. We have a very tiny tube-like structure of olfactory bulb, okay, which runs below the eye, okay, from the back to the front. And so this is one of the primary neurogenic region, the subventricular zone. Another neurogenic region is this part, which is the a small part of the hippocampus. Again, hippocampus, we just discussed that it is necessary for the learning and memory process. So this V-shaped structure in the hippocampus called dentate gyrus structure. Okay? And this dentate gyrus has, again, dividing cells which divide and get incorporated into the local circuit. And the Altman's group, they looked at the neurogenesis in this dentate gyrus of the hippocampus. So when the people studied this migration pattern along with, like we also studied the migration pattern of these subventricular zone cells all the way to the olfactory bulb, is a very stereotyped migration pattern. As one cell slide upon another, the third cell slide upon the second one, like that, they move forward. And the migration is very, very rapid, okay? But this migration now is, we know that is assisted by blood vessels, because blood vessels, so they use the blood vessels as an anchoring substrate onto which these cells remain, and then they migrate forward, one top of the other. So when we started working on this, what we did is you take a newborn mouse pup and you inject green fluorescent protein into one half of the hemisphere, and they then give electric pulse. When you give electric pulse, the GFP gene gets incorporated in the subventricular. So here, if you see here, this very bright area where the injection was done, this is the ventricle, okay? And then electric pulse was given, and the DNA gets incorporated within the cells, dividing cells, and then starts expressing this green fluorescent protein. And if you follow them few days, you find that these cells indeed they migrate through this tiny pathway, goes to the olfactory bulb, and then deviate out. Just switch 90 degree and then go to the different part of the olfactory bulb. So I'll show you how the migration happens. So if you see, this is the way the migration. So most of the cells, they're migrating from this end to this end. Some of the cells can migrate backward, stop at point of time. Some of the cells can just wander around, okay? And then again starts moving forward. So another group, they studied this kind of a migration pattern, and they found that some cells can, any cell can at one point of time move linearly, or then next moment it can stop, next moment it can wander around, again it can start moving linearly. So it's a kind of a very random kind of movement, but ultimately they anchor onto the blood vessels. Here the blood vessels are not visible, and then move from the zone of origin to the olfactory bulb. Okay. Now I said that the second neurogenic region is the hippocampus. Hippocampus is somewhere here if you look at brain from the side, and this is the dentate gyrus. This V-shaped structure is the dentate gyrus. Okay? And in this dentate gyrus also, the main dividing cells are again clearly in nature. So they are called the modified radial glial cells, or type 1 cells, like the cell here, which is present here, okay, the green cells here. And then they keep on dividing, and the cell changes its characteristics over time and ultimately forms the mature granule cells. And they get incorporated in the granule cell layer, which is somewhere here, okay? Because they connect to the CA3 region through mossy fibers, and then this CA3 then connects to CA1 region. So this is the neural circuitry. And if all the time these new neurons are being synthesized. Now, when we see in terms of numbers, in case of human being, as we are not using the our olfactory system very much, is not more, but our hippocampal neurogenesis is pretty much equal to the mouse system. Okay. But this neurogenesis reduces with aging, and that is the main problem with aging. And if you see this graph with increasing age, okay, this is experiment done in mice, but the same thing happens in the human being, the number of neurogenesis drastically goes down. But so if you have some cognitive defect, you will see that doctors say that if you exercise, etc., it helps. And that is true. Okay, a person who is old person who is doing Sudoku, it will help, because this is governed by something called a cognitive neural reserve hypothesis. So whatever neuronal loss is happening with aging, that gets compensated by the existing neuron being acting very, very efficiently. Okay, so it's a compensatory mechanism, you may say. So if you have, there are experiments which are done that if you keep an old mouse in a normal condition versus in a enriched medium, enriched condition environment, that is, you have some toys, etc., kept there, and you put the mouse for physical exercise for us, if a person runs, okay, or or walking every day, so the neurogenesis or cell proliferation increases. This is known, okay, and that is helpful. So why with age the neurogenesis goes down? It goes down simply because the cell cycle length increases. There are DNA damage that sets in, which starts reducing the efficiency of the cell proliferation. The level of growth factors goes down. That is also another factor of reducing the cell proliferation. So there are multiple factors which reduces the cell proliferation with age. One of the important factors is stress. Okay, it has been seen that the cortisol level, which is a stress hormone, goes up in the older people, and the number of cortisol receptors also in the neurons goes up. And this stress induced, or that's why, in case of depression, there is a huge neurodegeneration that happens. So stress mediated neurodegeneration is also very, very prominent in the older people, which leads to the reduction in the neural number. So if we look at the disease process, there are several diseases. In some cases, we have new neuron synthesis like Huntington's disease, stroke, traumatic brain injury, increase in the neurogenesis, adult neurogenesis. But in diseases like Parkinson's, Alzheimer's, we do not have that kind of situation. Okay. So we worked with this traumatic brain injury. I'll just briefly tell you what happens in traumatic brain injury. So if you look at a brain cartoon from the side, or a brain section from a side, this area we suck up some amount of the cortical tissue, and then we see what happens to that, whether the brain itself has some healing effect or not. So what we saw that the new neurons which are synthesized in this subventricular zone, some of them, they do not follow the normal pathway. They just deviate out or migrate out of this normal pathway and start moving to the site of the lesion. If I see in this structure E here, you see these green cells are the neuroblasts or neural progenitor cells. They come out of this migratory stream and then go towards the neural injury area. Okay. And we have seen that here also the migration is on the blood vessels. Here the red are the microblood capillary. These are the high resolution confocal images. And you have the neuroblast cells, they're just crawling up along the migration. But we also saw that the intervasculature, okay, which is normally like a very reticular zigzag vasculature in case of a normal brain, after lesion, the vasculature becomes more parallel in nature, and a restructuring of the vasculature happens. New angiogenesis or new vasculature forms, and that are directed towards the lesion site, and that helps these new neurons to go more efficiently to the site of injury. But unfortunately, we have seen that they can go up to that point of injury, but then they differentiate into some other types of cells. So ultimately, the number of functional cells going to the site of lesion is very, very minimal. Okay. Similar studies were done in cerebral stroke or cerebral ischemia. So when a cerebral stroke happens, it primarily affects this area, the close to the ventricle. Okay? This area is called the striatum area, okay, where there are a group of neurons. They are called medium spiny neurons. That's very tiny neurons. They have lots of processes, and they're very, very important and also vulnerable to any kind of oxygen deficiency. So these cells degenerate or die. So when in this condition, it has also been seen that the new neurons which are generated at this subventricular zone, they can deviate out and then go to the site of injury. And when people looked at it more closely, it was found that again, new angiogenesis takes place, new growth factor upregulation takes place, like several types of growth factors. And then these blood vessels act as a railroad for these neurons to go from the site of generation to the area where the neurodegeneration has takes place. Unfortunately, here also it has been said that most of these neurons die, and only 1 to even 0.1 to 0.2% neurons, they get integrated into the existing circuit. So these things are promising, but they cannot do anything on their own. So people work on transplantation biology. So they take healthy cells from other region and put it in the site of injury, and then see what happens. It has been seen that if suppose today a person made an accident and we do a transplantation surgery, if we do it immediately, it will not work. If we give some gap, 3 to 4 days gap, and then do the surgery or transplantation, then it will work. Why? Because immediately after, there are a lot of immune modulators that are present, which also kill the incoming transplant healthy cells. Okay, so if you give a gap, then the transplantation works better, and then they can get into the existing circuitry. We studied the Parkinson's disease. I'm sure many of you know what is Parkinson's disease. The neurons, the dopaminergic neuron, which is generated in the midbrain, specifically from the substantia nigra, they go to the striatum. But here the neuron degenerates, so dopamine level goes down, which leads to Parkinson's. We do not have much medications on that. So people turn out to the stem cells, because long-term medications has other side effects, something called a dyskinesia process. Okay, so what stem cells can do, you take stem cells, you put different factors, okay, different proper proteins into that culture dish, and then guide that stem cell to go to a certain lineage. It can form a neuron, specific type of neuron, it can form a glial cells, everything, okay, based on what kind of molecules you are throwing into the culture dish. So when we did this, we started with mouse embryonic stem cells and tried to generate this dopaminergic neuron in vitro, that is in the culture conditions. So this is just to show you all these red cells are the dopaminergic neuron, which were initially the neuronal stem cells, or not embryonic stem cells. And it takes around 1 to 2 months to do this entire culture process. And then you take this neuron and transplant, inject into the site of injury. So you can, so this is the area where we used to inject in the mouse. So you can actually create a Parkinson model in the mouse and then inject these things. And what we saw that once we injected these cells, these stem cells within the substantia nigra, they can make their way to the striatum. So all these green things are the extending neurons starting from the substantia nigra going to the striatum. So this is the picture in the striatum area. This is the picture in the initial origin area. Okay, and they form functional connections, which you can study using electron microscopy. They form myelin sheaths also. So things look promising by doing transplantation biology in case of neuronal studies. Another important thing that people now do is or try to understand the role of induced pluripotent cells. I'm sure you have heard about these induced pluripotent cells. The idea is you take the cells from the patient itself, from another source, okay, which are already differentiated cells. Then you, a process called de-differentiation, you again make them pluripotent stem cells and then channelize to a certain cell type. Okay, that way you can do something called autologous transplantation. So Shinya Yamanaka and John Gordon got Nobel Prize for talking about this, or for establishing these induced pluripotent cells. And now these iPSCs are being extensively used for therapeutic purpose. We still do not have any kind of a report where iPSCs are directly put and then they can recover, etc. But mostly nowadays iPSCs are getting used for disease modeling. For example, in case of Parkinson's disease, so if you take a skin tissue from the patient, okay, and then make them de-differentiate using this different factors and try to differentiate them into the dopaminergic neuron or astrocytes. So what experiment, recent experiment, I'm just telling you one. You have taken a healthy iPSCs from a healthy person, you take from a healthy person, you take iPSCs or make iPSCs from patient skin cells. So you take the skin cells, use these factors, OCT4, KLF4, etc. They are all transcription factors. And then you make them pluripotent stem cells, okay? And then differentiate these pluripotent stem cells from the healthy individual or patient into dopaminergic neuron or astrocytes. Now, when people did a co-culture, that means they first put the astrocytes, on top of that, they put the dopaminergic neuron. So if you take the astrocytes from the defective from the patient iPSC and put a healthy dopaminergic neuron, you get a defective dopaminergic neuron. This suggests that the astrocytes, they actually regulate the fate of the dopaminergic neuron. Okay, so if the astrocyte is not healthy, a healthy dopaminergic neuron can become unhealthy. Okay, on the contrary, if you have a healthy astrocytes and you take a dopaminergic neuron from the patient iPSC, the amount of defect gradually goes down in those dopaminergic cells. So this suggests that how in an in vivo system might be the astrocytes and the dopaminergic neuron interact. Okay? So this is how disease modeling are being done. Similar kind of disease modeling are done in case of Alzheimer's disease also, but we need to understand one problem here. Suppose if I take a cell from a person, suppose the disease is because of a genetic error, a mutation in certain gene. Now, if I take a iPSC, if I take a skin cell, make iPSC, it will still carry that mutation. So you will not get a healthy cell for transplantation. So what you need to do, or what is being done right now, is to also incorporate gene editing process using CRISPR-Cas technology. Okay, so you make the iPSC, do the gene editing, so rectify the errors that was present originally in the cells, and then make the cells of your interest and then transplant them. Okay, so this is how the entire therapeutic, cell-based therapeutic is going ahead or moving towards to ameliorate different diseases or neurological diseases where there is extensive neurodegeneration. So with this, I would like to thank you all for giving me the opportunities, and if there is any more questions, please feel free to ask.
For the amazing session. Thank you so much, sir. It was very informative about the neurogenesis and the related aspects. So now participants can ask questions through their live chat box. So sir will answer all these questions. And the feedback form of the this particular lecture is available on the live chat box. Participants, please be sure to fill them. Sir, may we conclude this session? I think there is no more questions.
I think, yeah.
It's a pleasure to have you, sir. Thank you so much.
Thank you. Thank you.
Are you there, sir? Can you hear me? Hello, sir?
Now we can start our next session. Ma'am is here.
Okay. Now it's a time for the second lecture series. Uh, by Professor Vidita Vaidya. Now let me introduce our next distinguished person and speaker for Professor Vidita Vaidya. Welcome, ma'am. She's currently a neuroscientist and professor at Tata Institute of Fundamental Research, Mumbai. She holds PhD in neuroscience in Yale University. She even holds two postdoctoral research, one at Karolinska Institute, Sweden, the other from University of Oxford, UK. After successfully accomplishing her degrees and doctorate, she was awarded the Shanti Surad Bhatnagar Award in the year 2015 for her notable and outstanding research in medical science. She earned fellowship from International Science Academies. She was honored with many other awards that include National Bioscience Award for Career Development and Nature Award for Mentorship in Science. She was a senior research fellow at Wellcome Trust Overseas. For a period of 5 years, she was an associate member of Indian Academic Sciences. Having bagged all the prestigious posts and being specialized in a number of works, she got opportunity to get featured in the book Leelavati's Daughter. I am gladly welcome her for the speech.
Thank you. Thank you. Thank you, Niduna.
Thank you, ma'am.
Should I share my screen?
Yes,
Let me just
you can,
try
ma'am.
that. Yeah, hold on. Let me see. It's going to work. Can you confirm if you can see my screen? Is it
Yes,
visible?
ma'am, of course. Yes, of course, ma'am.
Yeah. Okay, great. And if at any point, either because of connectivity related issues or something goes wrong with the screen, please stop and interrupt so that I know that there's been a problem. Yeah.
Well, sure, ma'am.
Thanks so much. Okay, so good morning, everyone. Happy New Year. And hopefully this year is going to be a better one than the one that we had last year. Certainly with the discovery and the invention of the vaccine-related technology that we are seeing, hopefully we will see some sort of a control happen on the coronavirus pandemic. So what I'm going to tell you about is perhaps relevant given what we've all lived through in the last year, which is what happens to your brain when it is under stress. And really, the fundamental question that one can ask oneself is that when one is stressed, does one adapt to the stress or not? And that really is a central question in a sense of how we handle and cope with stress. So, first, let's start by defining stress. Everyone hears this word all the time. You will usually say it like, I am so stressed. I've had such a stressful day. It's been a stressful month. The entire planet is walking around saying it's been a terribly stressful year. 2020 has been so stressful. But what does this word stress actually mean? And can we biologically define it? So, stress can be defined as anything that disrupts the homeostatic balance of your body. So, for example, if you're sitting out in the blazing sun the entire day and you end up sunburned or end up with a sunstroke, that is a stress. If you're sitting in your, sitting at home and worrying about your exams that are going to happen 1 month later, that is also a
Ma'am,
stress.
can I just ask?
Yeah.
Are you sharing your screen right now?
Yes, I am sharing my screen. Can you see it?
But no, we can't see it.
Okay, that's good to know. For some reason, okay, I did share the screen and said share screen. Let me see if I can share my entire screen. And if that helps, let me just see. Can you see it now? Is it visible now?
No, ma'am.
Okay, that's a bit surprising. So I'm hitting the share screen button on this, at which point it says, share, choose what you'd like to share. And I've said share my entire screen and I've hit share. And for whatever, let's see, why has it lost the permission to share the screen? Okay, hold on a second. I'm going to try to see what the issue is. Let me just quickly try and see. Working. I usually use Zoom, so that maybe it's this particular platform. Give me a second. I'm just going to try to see.
Okay.
I'm just going to try and see if there's something with the system preferences that will allow me to fix this. Security and privacy. Sorry about that. Give me a minute. Let me just try and see.
Dear participants, if you have any questions during the lecture, please type them in the chat box. We will discuss at the end of this lecture. Sure. Thank you.
Hi, I'm back. Let me try again and see if it works this time. Let's see if that works. Are
Yes,
you able to
ma'am.
see my
It's coming.
screen?
Yeah,
this time
yeah.
around? Okay, that's good. It
It's
was
fine.
just a privacy issue with the way it was set up. That's fine. I'll just yeah. I'll just okay. Can you still see it? Is it okay? It's clear.
Yes, ma'am.
Hello?
It's clear. Yeah.
Can you confirm you can actually see my screen?
Yeah. I can see it.
Okay, great. Just
Yes.
confirm so that I'm sure that it is visible. All right, great. So I'll then continue. So as I was just describing, that stress is any condition that disrupts your homeostatic balance. For the last 2 seconds, when I've been trying to figure out how to get back connected, my body would have gone through a minor stress response, a little one. But nevertheless, I would have produced a little bit more cortisol, secreted it, trying to figure out how to get connected. The organizers of the meeting would have worried, oh no, what happens if our speaker can't get connected back on? And they would have also had a slight minor cortisol response. So our body is ideally equipped to deal with acute physical stressors. So how should we think about this? Perhaps the best way we can think about this is through the evolutionary window. So think about it's just 10,000 years or so since we started agricultural farming and farming communities and villages built up and then cities. But long before that, we were a hunter-gatherer society. And so, what were your major sources of stress? If you couldn't find any food to eat, you would be stressed. And you also wanted to be sure you didn't become somebody else's food, right? These are the two big things that were the major worries of your life, at least more than 15 to 20,000 years ago. But our system was beautifully equipped to handle that. And we were well equipped to sort of run after something or climb up a tree and pluck berries, or run after some prey, catch it, cook it, and make food. So these were the kinds of stresses we were dealing with. We also probably had to run away from other large big cats and mammals that might attack us and make us their food, right? Facebook or Snapchat or TikTok, and you've posted something and you're waiting to see how many likes you get. And many of you may care or not care, but sometimes it starts becoming something that matters to you, which is the positive reinforcement we seek from the world. If you just seek it and that's where it ends, it's fine. But if you start getting stressed about it. Let's give another example. Let's say you're sitting in a classroom and you have a teacher whom you really like, but you've got this feeling that this teacher doesn't like you. And you're convinced that this is going to impact your grades, your recommendation letters, and now you start worrying about it. Then you go home, you talk to your friends, you talk to your parents, you say, I think this teacher really just dislikes me. And then this becomes an actual thing in your mind. And over a period of time, you start worrying about your performance in that subject. You start worrying about your relationship with that teacher, advisor, and this starts consuming your mind. And this is actually stressing your body out. And yet, it's not an obvious stress like the stress of, not eating and trying to find food, or, the kind of obvious stress of if there's a cheetah running after you, that is a genuine stress, right? So you worry about that. But these are also genuine stressors. And these are the real stressors we have. How many of us have to worry about where food is going to show up on our plate? It somehow shows up. Now it shows up from a grocery store or from a restaurant or from a Swiggy app. You're not walking around in the savannah trying to catch your food. You're also not walking around in the savannah avoiding becoming somebody's food. So if you look at this hierarchy of stressors, most of your time is spent on that third category, which is psychological and social stress. And what is your stress response? Your stress response will be your body's attempt to restore homeostatic balance. I have yet not defined homeostatic balance for you. I've just told you that stress can be a plethora of things. It's many, many different things. Your stress and my stress may be different. There are many youngsters who like the idea of doing bungee jumping. I can't imagine why anyone would tie a rope around their ankles and jump off anything. I mean, it seems horrifying to me. But it's a entertainment for some and form stress. So you say something is stressful to another person. Really, it's eventually how you handle that stress, static balance. Responses are adaptive. This category on the left-hand side. The goal is after the stress is over, or even very soon after the stress starts, you start handling the stress. And because you handle the stress, you come back to normal or you come back to baseline. But not all stress responses by definition are always adaptive. More often than not, most of us will adapt to stress. But some stresses can be maladaptive. And when they're maladaptive, you don't return back to the normal baseline. In fact, you often return back to some sort of a disrupted homeostatic balance, and that actually adds something called an allostatic load, a load of coming back to something different from normal. You're coming back to something, but it's not baseline, okay? And when you have this kind of a maladaptive state, and you have this lasting for long periods of time, then you start precipitating and worsening stress-related disorders. And what are some of the stress-related disorders? Depression, post-traumatic stress disorder and anxiety, cardiovascular disease, stroke, hypertension, ulcerative colitis, irritable bowel syndrome, immune dysfunction. This list is large. In fact, there's pretty much no disease that is not worsened by stress. Even though it may not be caused by stress, pretty much every disease gets worse if you are also stressed at the same time. So stress-related disorders are a major concern of our times. Now, why did I put this cartoon up? But part of it is to make sure you're paying attention to me. I mean, this photograph is an amazing photograph. It's just to give you a sense that you have two animals here, both of whom are under stress. Acute physical stress. The deer is under acute physical stress of running away, and the cheetah is in the acute physical stress of actually catching the deer and eating it, right? So both of these animals are under acute physical stress. And acute physical stress, what do you want to do? Okay, you want to perfectly adapt to it. And how do you adapt to it? So the deer is going to, suppose the deer has been eating some grass, and the deer was digesting this grass, and now along came this cheetah. Now the deer has to run at full speed. It doesn't have time to digest the grass right now. It has to use its muscles to run as fast as it can, right? So what is it going to do? It's going to shut down the digestive pathway. It's going to divert the blood flow to the muscles. It's going to say, okay, all the blood and sugar that I have in my system, all the glucose, let me send it to my muscles so that my muscles can move as fast as they can. So that's what you're going to do. The same thing for the cheetah. The cheetah is also not going to worry about digesting its previous deer. That deer is history now. It's going to have to catch the next deer, perhaps for itself, perhaps for its babies. And so it's also going to be mobilizing glucose entirely to the periphery, which is to the muscles. At the same time, these animals are going to worry about the possibility of injury. And so immune systems are primed for injury related responses. And so the immune system is in some ways being primed in an expectation of something. You're also shutting down reproduction. So you're going to shut down the hormones that are associated with reproduction. You don't want to waste time thinking about reproductive success when you actually have to worry about survival. And so these systems are really beautifully adapted for this. You said if you keep having to do this, you can't shut down digestion forever. That's not a good thing. Physical stress, shutting out your digestion is not going to be a great thing. So you're going to have to adapt to that. But now you imagine if you do this, now let's just take this photograph and look at the deer and the cheetah. And imagine that you are the deer, except change the entire context. Most of you are college students. So I'm going to imagine and tell you that you are that deer, but you're sitting in a classroom full of people. And your best friend has just decided to stop talking to you because you all have had a horrible fight. The same hormones are going through your body that are going through this deer's body because you are traumatized, you're upset, you're angry, or emotions are you've also eaten food, but your digestion is now deciding to shut off because it's decided, oh my God, this body is under stress. So let me now not worry about digestion. Let me send glucose to the muscles in case this animal, which is you in this case, needs to handle the stress. But this is because of your best friend fighting with you. Maybe you're waiting for an exam result. And as you wait for an exam result, you do the exact same thing. So our systems were beautifully adapted for acute physical stressors, but think about the conditions in which we are using it now. And so now I'm showing you this photograph to tell you that, and many of you will recognize the building on the right-hand side. That's the Bombay Stock Exchange, and that's Dalal Street. And you see all these people staring at the Bombay Stock Exchange BSE, and their fortunes are going up and down with the way the stocks are trading. And look at the animals on the left. All three are stressed. The three animals on the right, which are human beings, are also all three stressed. Their responses are identical. They activate the stress response hormonal pathways. They secrete cortisol into the blood and at very high levels because the body is preparing for a stress. But look at the conditions on the left. Do you think they'll end quickly? Well, they will. Either that baby deer gets eaten or the cheetah gets attacked or something happens. It's not going to last forever. Look at the condition on the right. How likely is it that this is a long-lasting condition? Well, if you're a stock market trader and this is your job, this is your everyday life. And you're going to be doing this for months and years and years and years on an end. And yet your body is responding the same way as the animals on the left, where the situation is going to come to a conclusion very quickly. So now I want to tell you what killed us in the past and what kills us now. So look at this particular graph and look at what's on the x-axis. It's 1900 and 2010. Okay, so these are sort of the numbers of what were the major reasons for death. This is United States, US numbers. So it'll be slightly different for different parts of the world, but let's use this as a reasonably decent representation of the change from 1900 to 2010. What already you know is people are living longer. So the number of deaths, for these diseases, you see that life expectancy has gone up. In the past, in 1900, people didn't live to be in their 70s and 80s. That was not common. 50 was like a really long, well-lived life. But in 2010, people lived to their 70s, 80s, and 90s. And look at the causes of death. Look at the causes on the left-hand side. Infectious disease is a major, major player. On the right-hand side, it's cancer, heart disease, etc., etc. And of course, there is infectious disease, but it's still a small component. 2020, infectious disease will show up as a major component because we have 1.7 million people who died last year because of COVID world over. And if you just compare the numbers to 1918, 50 million people died in 1918 because of the Spanish influenza. So we are at the beginning end of this pandemic. And yet, in terms of our capacity in the last 100 years to handle a pandemic of this scale has changed so much because of biomedical science. Why am I showing you this? What I want to tell you is that the reason we die now are predominantly because of stress-associated diseases. And what is the stress pathway that I kept telling you about? Well, in mammals, here is the really conserved biological pathway. You perceive a stress, whatever that stress may be, it activates your hypothalamus, which secretes a factor called the corticotropin releasing factor, which is released to and goes to the pituitary. The pituitary secretes adrenocorticotropic hormone, which is again another hormone that makes it through the bloodstream everywhere in your body, acts on the adrenal gland, which sits on top of your kidneys bilaterally. So you have two adrenal glands. And the adrenal glands, the adrenal cortex and the adrenal medulla secrete cortisol, which is a hormone, and epinephrine, which is again another hormone or also a neurotransmitter very often. Both of these prepare your body for the stress response. Epinephrine will prepare your body for an immediate response. It's very easy for you to figure this out. If, for example, I ask you to quickly solve a mental math sum, okay? So suppose I say 110 multiplied by 215, and you quickly try to solve it. And then you measure your heart rate before and you measure your heart rate after, you'll see a small spike in your heart rate, small one. And it's usually quite reliable. Maybe you can do this as an exercise when you go home. Maybe, or you are already home probably because of the pandemic. So catch some two people in your house and say, I'm going to do an experiment with you, where you first measure their baseline pulse rate, then you make them do mental math and set them challenging sums, and not impossible to solve sums, but sums that will require them to use their mind a little bit, and then measure their pulse rate after they finish the sum. And you will see that the pulse rate goes up because it's a small acute short stress. And it gives you a nice boost in your heart rate. And all of you, when you're stressed, know that your heart rate goes up. Okay, so this is a pathway I told you about. It's the fight and flight response pathway, where basically the hypothalamus secretes corticotropin releasing factor, which acts on the pituitary, which causes a secretion of another hormone. Don't worry about these terms. It's not that important. Bottom line, what I want you to know is that we have a nice, beautifully engineered, evolutionarily conserved, nice stress response pathway, which we all switch on when we are under stress. And where do these hormones work? They work in many places. Like I told you, in the muscle, they will increase glucose transport. But in addition to the muscle, they're working in many parts of the body, including your brain. They're working on your digestion, they're working on your reproductive tract, they're working on your muscles, they're working on your heart, they're also working on your brain. So on the left-hand side, you see the human brain. On the right-hand side, you see the rodent brain. And the structure I want you to focus on is the structure called the hippocampus. It's called the hippocampus because in someone's fancy idea of the world, when they dissected it out, it looked like a seahorse. And hippocampus actually means seahorse in Latin. I know it doesn't look like a seahorse in this image, but trust me, when you dissect it out, it kind of does look like a seahorse, which is curled up on itself. So here's the rat brain, here's a human brain. For most of my talk today, I'm going to tell you about what we studied with the rat brain. So let's just focus on this. This is where the nose of the rat would be. This is where the tail would be. So this is the direction. And this is this really large structure called the hippocampus. It's also very large in the human, except that we have this big cortex which expanded. And because of our cortical expansion, the hippocampus looks proportionally smaller. Whereas we don't have that large a cortical expansion in rodents. So rodents have a smaller neocortex. Okay, so this structure, the hippocampus, which sits bang next to this almond-shaped structure called the amygdala, are structures that are very sensitive to stress pathways and stress hormones. And why is that? One of the reasons is because they express a ton of the receptor for those corticosteroids. So corticosteroids is the hormone cortisol, is a stress hormone. It's what you and I secrete when we are stressed. In rats, they secrete a paralog of it called corticosterone. These are all cholesterol-derived hormones. And this hormone will bind to the corticosteroid receptors, which are shown here. And you can see that the hippocampus, this is the brain, cut through, you can actually see these sections, very highly express the corticosteroid receptors. So the major target of the hormones in the brain. Okay, so when you get stressed, the hormone's going to secrete. It's going to go through the blood-brain barrier, and it's going to be obviously binding to cells in the hippocampus because they have a lot of the receptor. So this just tells you that. I don't need to tell you more than this. Okay, the other important thing I need to tell you is that the hippocampus has a very important job. By the way, the hippocampus is very important for learning and memory, for your recall of events, etc. Okay, if you listen to me well, and then you maybe a few days later want to describe what you heard to one of your friends, you're going to need your hippocampus to do it. First, you're going to need your hippocampus to pay attention and remember what I said, and then you're going to need your hippocampus later to recall it as well. It's a very important structure in the brain. I've already told you about the hypothalamo-pituitary adrenal axis. Sometimes there will be words and jargon thrown out. Don't worry about the jargon. The bottom line, this is the stress response pathway. I told you that the stress response pathway secretes glucocorticoids or corticosteroids or cortisol or cortisone. All these are the same broad name for stress hormone. So it secretes stress hormone. What I have not told you yet is that as important it is to secrete the stress hormone, it's equally important to shut it off. So there is a very nice feedback inhibition. So when you get stressed and you secrete the glucocorticoids, they actually go back and bind at the level of the axis and switch it off also. Okay, so there's a feedback inhibition. Much the same way, not only do they bind at the level of this axis, they also bind in the hippocampus. And the hippocampus is known to inhibit or switch off this axis. So this is a nice way of controlling and shutting off the stress response. Now what happens with stress is very interesting. I just told you that the hippocampus is responsible for shutting off the stress response. I also told you that the hippocampus has a lot of receptors that bind the stress hormone. It's beautifully designed for an acute stress. For very long-lasting, chronic, severe stressors on which you have no control, over a long period of time, if you keep exposing these neurons to the stress hormone, these neurons start dying or getting damaged. So imagine that there's a lot of stress. So you increase a lot of stress hormone. This stress hormone goes and binds to the hippocampal neurons and eventually destroys and kills them off. Because you kill off hippocampal neurons, you have fewer hippocampal neurons. If you have fewer hippocampal neurons, you will not be able to shut off the stress response pathway. So this is what is known as a vicious cycle. You keep doing this, eventually over a period of time, this will amplify and create a situation in which you cannot control and shut off your stress response. So now I want to tell you about adaptive and maladaptive stress. I told you that at the very beginning that most of us always adapt to stress. Okay. I presume that Bhaskar has already introduced you to the idea that there are new neurons born in the brain, of course, when you're developing and when you're a baby, but new neurons are also born in the brain when you're an adult, and in particular in the parts of the brain which have these neurogenic niches. One such place is the hippocampus. Guess what? When you're adapting to stress or you're handling a stress, you actually produce more newborn neurons in the hippocampus. Anxiety actually goes down. You improve learning. And you actually have an improved immunological response. And this is what's happening when you're adapting to stress. And that's a good thing. More often than not, we always adapt. So that's a good sign. And you all would know this, right? Being mildly stressed for an exam is good because when you're slightly stressed, you work a little harder. Your concentration is better. You're more focused. You may go and actually crack the exam or you do really well. And you're very happy with that little bit of stress. But when the stress becomes very bad, such that you haven't slept for weeks, you've been completely freaked out about the exam, then you see the questions and the questions become gobbledygook in your mind. You can't even answer anything. This is the zone when you've gone into the maladaptive. When you're very stressed for long durations and you can't control the stress, you can't switch it off, you actually reduce the production of newborn neurons. You get highly anxious. In fact, your learning and memory really suffer, and you also impair immunological responses. So here I'm summarizing some of the adaptive and maladaptive effects on the hippocampus. In the hippocampus, you can divide it as a trisynaptic circuit. You have the CA fields where the pyramidal neurons sit. You have the granule cell neurons in the dentate gyrus. And under stress, you actually, under adaptive conditions of stress, you actually promote the formation of spines and growth of dendrites. You actually give birth to more newborn neurons. In fact, you improve learning and memory through improving long-term potentiation. I haven't defined that, but suffice it to say it's a cellular basis of learning and memory. And in fact, these animals actually do much better on learning-related behaviors, and they do much better even when their anxiety is reduced. But if you look at maladaptive effects of stress, the same trisynaptic circuit, you damage the neurons. In fact, you reduce dendritic branches, you remove spines. In fact, newborn neuron production goes down dramatically, and there are actually far less new neurons born. At the synaptic level, you end up with actually a shift away from learning and memory, and you do not facilitate long-term potentiation. Animals don't learn well, and they are highly anxious and show pro-depressive like behaviors. So to come to the second and the last part of this talk, how am I doing for time? How much time do I have? Just wanted to check. You guys can hear me, right?
Yes, ma'am.
Yeah, audible, and the slides are still visible. That's good. That's good.
Yeah.
Do I still have about 15 minutes more for the talk? How am I
Yes,
doing for
ma'am.
time?
Yeah.
Yeah. Okay, great.
The
Okay.
breaks will start at 1 o'clock.
Okay, perfect. That's wonderful. All right. So let's ask this question. I told you about adaptive effects. I told you about maladaptive effects. I told you about the response pathways. I told you that you will adapt well most of the time. But maladaptive is when you can't control the stress. It's too severe. It's too long lasting. It's something that you just really can't switch off very easily. This often happens for psychological and social stressors. Can also happen for physical stress. But very often, physical stress, there is at least a solution that can be employed if possible. Psychological stressors, it may not be as easy to apply that solution. What I have not told you is, does timing matter? Does it matter when you're exposed to the stress? When you're a child versus when you're an adult? And here you see baboons on the left-hand side. Does it matter which end of the spectrum of age you are? What happens to the stress responses then? And this is something that my lab is deeply interested in. We're very interested in how early environment, that early window, shapes the development of stress responses. So, in fact, your stress responses develop when you are young. How you fine-tune your stress responses is very often programmed by the kind of environment you're exposed to. In animals and rodents, in the first 3 weeks of life, they're actually forming and shaping and reorganizing their stress response pathways. So, for example, if you're exposed to a non-stressful environment, a very enriched, happy, generally good environment, then you develop a modest stress reactivity. In fact, that reduces your risk for disease, not just then, for the rest of your life. If, however, on the other hand, you have had an early environment in which there has been profound stress, and you have had adversity and challenges, and many of those challenges have not been easy to resolve, it's associated with increased stress reactivity and increased risk for heart disease, type 2 diabetes, alcoholism, the entire spectrum of psychiatric disorders, aging, etc. This is why timing matters. In adulthood, when you're exposed to stress, you're not setting up the stress pathway. It's already set up. So you will respond based on the way your pathway was set up in many ways. In the early window, you're shaping that pathway. And so stress in the early window can have very long-lasting consequences. It programs lifelong behavior patterns. It can set up a lifelong vulnerability to stress. For example, here in this image, and this is work that's come from the Nemiroff lab, you can see that animals exposed to lower maternal care actually have more cell death. Neurons are dying because they've suffered neglect. In this species, please remember that the primary caregiver is a mother. The minute we move over to other species and primates, it becomes a much more complex caregiving society. So it's not enough to simply say that all the responsibility of care is on the mother. The responsibility of care is on mother, father, aunts, uncles, relatives, caregivers, teachers. It's a much more social context. In fact, our babies are born very, very immature, and for the first decade of their life, they depend on our species to nurture them well. And the quality of nurture in that first decade has lifelong implications. So when this has been studied in animal models, work coming from the Meany lab at McGill, what the lab did is they set up models in which there are high caregivers versus low caregivers. And this is based on just observing the quality of maternal care. When you look at the babies born to mothers, rodents in this case, that give high maternal care, they have low anxiety and they have a very strong feedback control of the hormonal stress response. They switch off the response really quickly. On the other hand, babies born to mothers that are low quality caregivers have very high anxiety in adulthood, and they have a poor feedback control of their hormonal stress response. And why does this happen? Because the nature of care that they receive directly determines the number of receptors they will have in the hippocampus using an epigenetic based control of expression of this particular gene. And so if you have more receptor, the minute the receptor binds the hormone will bind, activate the hippocampus will shut off the stress response pathway. If on the other hand, you have very little receptor, it takes much more hormone to actually switch on the hippocampus to switch off the stress response. So you have poor feedback control. And this is regulated by determining the number of receptors that are expressed in the hippocampus. So high caregivers end up with higher amount of receptor expression. Low caregivers, babies born to low caregivers, end up with a lower expression. Because of this, the high caregiver babies actually end up with a very strong feedback inhibition of the HPA axis. The low caregiver, the infants that receive low care end up with a poor feedback control. What this will mean is that the brain of these animals will see much higher levels of corticosteroids compared to this. And you will see this in the next cartoon here. In this image, in this graph, work coming again from the Mini lab. Here is the pre-stress condition, and you're measuring the amount of plasma corticosterone. This red line indicates 20 minutes of stress. Both high and low end up giving you an increase in corticosterone, but you can see how quickly it reverts back to baseline. Here's the integrated corticosterone. Here's the low caregivers. They give a higher peak, and it takes them much longer to come back to baseline. And so this ends up meaning that the brain of the babies that had low care end up seeing much higher levels of corticosterone for longer periods of time compared to those that were given quick and high quality care in early life. Here's another piece of work. This is work coming from my lab, from Deepika Suri, who was a graduate student in my lab. What Deepika was interested in asking was, what happens when you've undergone early stress? So this early stress involves a paradigm called maternal separation, where the pups are taken away from the mother for 3 hours every day from postnatal day 2 to 14. And they're really cute at this age, by the way. And here they are as a cohort. They go away from their mother. They come back to the mother 3 hours later. 3 hours is a very short separation. It's not a very long separation. But what happens in the laboratory context when you do this is it deteriorates the quality of care that the dam provides to her pups. The licking, the grooming, the archback nursing is far less. And so it becomes a model for fragmented maternal care. The maternal care is disrupted. And these animals, when they grow up, show a whole bunch of changes. So what Deepika, who's a PhD student in the lab, and Vandana, who was a master's student, decided to ask was, okay, let's make these animals go through maternal separation, and then we will check them when they are 3 weeks old, when they're really juveniles. We look at them when they're 2-month-olds, when they're young adults, healthy, full-fledged, sexually mature adults. And we will look at them when they're 15 months old, when they're fairly middle-aged. And we will measure cognitive behavior. We looked at structural changes in the brain. We'll ask what are the molecular and epigenetic markers that are associated with this. And then we'll ask, okay, if we see changes, can we reverse them? And that's what Deepika and Vandana did for their respective thesis work. And what was fascinating is that Deepika and Vandana showed that actually, when you look at them at 2 months of age, and this is looking at them on a learning task where the animal has to learn to find a hidden platform buried underneath milky water. And so the animals initially get put in the water, they swim around, they can't find the platform, but eventually over time, they really learn very quickly where the platform is. And they figure this out based on cues around them. Just like you will figure out a way to go to your college. If the first time you won't find it easily, but after 1 year of going to your college, you almost close your eyes, you can give directions and you can get there, right? So animals are also really super smart. They also very quickly learn where the platform is hidden. And they figure out by day 4, everybody figures it out very quickly. But they have to be trained. So they go through 3-4 training sessions a day. And during training, what you find is that the maternally separated animals, the ones that went through stress, actually do better. So this was really surprising. We were quite surprised, and so was the field. When we published this work, it was not very obvious. We would have thought that the animals that went through stress would not learn as well. In fact, they did exactly the opposite. They learned even better, and they were much faster. You can see they were faster on day 1 across 4 trials. By the 4th trial, they're outperforming the controls. So this is the controls. These are the animals that went through stress. So they learn, and they learn better. And then when you ask them to remember it, like, 1 day later or 10 days later, they all remember it really well. So everybody's learned it. They're all remembering. Now you look at them when they become 15 months. Okay. So this is another cohort that has become now middle-aged. All those benefits of learning better are gone. Okay. So later on, when you're a middle-aged animal, you don't really actually show any difference on the learning or on the short-term recollection. But on the long-term recollection, you are doing worse. So if you had a history of early trauma, you will forget. Actually, you will not remember really well. You learn, but you don't remember it. Okay. Now you're looking at a different behavior. This is a behavior where you have to identify an object. These two circles are an object. Actually, it's usually a flask or it's a bottle filled with sand and then it's capped and put inside that. But for cartoon purposes, they are shown as circles and squares. Okay. So on the first day, the animal goes into the box and then it sees these two identical objects. And obviously, it likes to check out new objects. So it goes and checks them out and it plays with them and it looks at them and all that. The next day, you say, do you remember that you saw this round object before? Well, you're not really asking the animal that. You're watching the animal's behavior. You're trying to figure out if the animal actually remembers this. And how do we know it remembers it? Because all animals like novel objects. So we simply measure how much time it spends with this object versus this object. And if it remembers this object, it will spend most of its time with this new object. And sure enough, 2-month-old animals that went through trauma are just like controls. Controls or early stress, they learn very well. And if 7 days later, we give them another new object and the same familiar object, it still remembers, oh, I've seen that before. It doesn't spend time with what it's seen before. It likes to explore new things, so it goes and spends time with this object. So whether you've been stressed in early life or not, you will learn. And on this task, which is a stress neutral task, the previous task had a stress component to it because the animals were swimming. So you learned better on a stressful task, but on a non-stressful task, you're as good as anybody else. Okay, so whether you had stress in early life or not makes no difference whatsoever. But look what happens when you get old. When you get old, which is 15 months of age, you just can't learn. You just can't learn. Even when you've seen this object day 1, day 2, day 7, you still are not able to remember it as well. The controls are remembering it really well, but if you've had early trauma, you're showing cognitive decline. And that cognitive decline is quite dramatically obvious. Okay. So what does this tell us? This tells us that it depends when you look. In fact, if we had only looked at adult animals, we would have said, oh, actually, when you go through early stress, you do better on stress-related learning tasks. And you do better on many things. And yes, you do. You do better on certain tasks because your system is trying to adapt. But you pay a cost for it later in life when you're middle-aged. And this is true not just for behavior, it is true for many things, which I've just summarized here. Initially, you see an improvement in neurogenesis, an improvement in trophic factors. In adulthood, you still see improved learning, improved function on learning, improved BDNF, a bunch of other changes that are happening. But when you're middle-aged, that's when all the costs seem to show up. You show decline in progenitor turnover, you show cell death, you actually show a reduction in trophic factors, you show a failure in learning, whole bunch of things go wrong. And that becomes most obvious much later in life. And so why am I highlighting this? That what happens in a short transient window in early life can sometimes program lifelong behavior patterns. In fact, exposure to early stress can result in lifelong deficits in memory function, stress responses, and can even decrease life expectancy. This is not our work, but this is work, very elegant work coming from many groups that shows that life expectancy in humans has been linked to early trauma and early adversity. The question then is it all bad news? I mean, there are times we can't control what we've gone through in early life. There are times one cannot change those circumstances. So then what? Then one just prepares for all bad things happening later? No. Because the good news is that things are reversible. Things can be impacted. Foster care, adoption, stroking, enriched environment have profound effect on reversing these effects. I just want to end by sort of acknowledging my team. This is my lab. The two sort of central questions that my lab addresses are, how does life experience talk to circuits in the brain to actually shape behavior? And what are the neural circuits and pathways that underlie antidepressant action? In my second talk today, I'll be talking about the second topic, which is talking about drugs that affect the brain. Today, at least in the first talk, I've told you about how experience and what you live through can change the way your brain circuits actually function. And I should just end by acknowledging my collaborators on this particular study, which I talked about, which Deepika did. Sanjeev Galande is our collaborator at INSA Pune. And our funding comes from TIFR. This is the TIFR campus, southernmost tip of the beautiful city of Mumbai. And my lab is actually right here in a small little hut. And I am going to stop sharing my screen and take questions. Yeah. All right, we can take questions.
Thank you so much, ma'am, for the amazing session about how life expectancy, trauma are related, and all the aspects. So, can we go ahead for the question and answer session, ma'am?
Of course, absolutely. I look forward to the questions.
Okay, ma'am. The first question asked by Dr. Pishardi is that how long it will take to change hippocampus to change permanently?
Okay, so it's an interesting question. I mean, the good thing is that there's nothing permanent about these changes, and some of them are reversible. So to give you a sense of how dramatic these changes are, they can happen across hours. So spine changes happen within the hours, minutes time scale. You are constantly adding spines and removing spines from your hippocampus. Hopefully, if you're paying attention to me today, you might have added a few spines or you might have removed a few. So that's constant. That kind of change happening in your brain is happening at all times. If you're talking about more sort of persistent changes, like if you kill a neuron, then replacing it is not that easy. Those are far sort of severe changes that can last for years and that are difficult to reverse. So I don't think in the nervous system you should ever think in terms of permanent change. Unless you're killing a neuron and getting rid of it, there really isn't any permanent change. Many things are constantly changing. And the nervous system is a very plastic structure in that sense. So I hope that answers it.
Okay. And he also add to those questions that what will be the intensity of the damage to hippocampus?
It depends on the severity of the stress. So very severe, uncontrollable, unpredictable stressors can result in cell death. And those are really permanent changes. When you kill neurons and large numbers of neurons, then those are not easy to repair. But that happens with only very severe, very, very traumatic stress. The best way I can describe this is human beings are amazing, and so are all other mammals. We have a remarkable capacity for resilience. We survive incredible traumas. If you think about what we as a species sometimes do to each other, example being the Holocaust. 6 million Jews were killed. If you think about the Bengal famine in India, 3 million Indians died, utterly preventable deaths. Could have been prevented so easily if Winston Churchill had simply released the food that was in the granaries. We lost 3 million Indians to the Bengal famine. So traumas of those scale also people recover from. People recover from severe traumas. It's not that as a species, we are adaptable. We do handle trauma, but if it goes beyond a certain breaking point, then it becomes very, very severe and very difficult to repair. And if you look at our current world and the crisis that we have with reference to mental health, it's telling us that we are pushing the system to the point where there's not very easy room for recovery.
Yes, ma'am. So, ma'am, the other question is, what will be the sensitive period in infants to happen this irreversible damage to hippocampus?
So again, I want to avoid the word irreversible. I'm just going to go with saying that, yes, there is the possibility of damage. The critical period varies from animal to animal. So for rats and mice, it's the first 2 weeks of life. For humans, it's been debated whether it goes on all the way up to 15 to 20 years of age. Certainly, the medial prefrontal cortex connections to the rest of the brain continue to form up till around about age 25. So that circuit is still shaping and forming for the first quarter of life. So I would say the first decade in humans is absolutely critical. Absolutely critical.
Yeah. Okay. Then comes the next question. What is the observation of the receptors, particularly long-term effects, when a caregiver of a different species is provided? For instance, the case of adoption of a dog's puppy by a human.
I love that question. I think it's a terrific question. I don't think it's been studied in the case of a puppy, but I can give you an example of the way it's been studied in rats and mice. So let's say rats and mice are going through a trauma, and if as a human being you sit with a brush, and this is work coming from Stefania Macari's lab, and you simply stroke the rat, okay? You sit with a brush and you wet the brush and you just keep stroking the rat. Well, that stroking has profound positive effects on the brain. And that's pretty dramatic. So I can imagine that, yeah, when you get a puppy and a new puppy in the house and the puppy has been taken away from its mother and its siblings, I'm sure that's quite traumatic for the puppy, because it's used to being around, at least, well, till it's a certain age, around the mother and the siblings. But you will also see that the baby, initially the puppy will cry, or the puppy will really be not very happy, and then it bonds with the humans. And of course, there's no question that that bond is a very tight bond, and pets of that nature provide profound support to the humans, and the humans are also providing a lot of affection to the animals. So I bet there are changes. I don't think anybody's looked at it, but it's a great question.
Yes, ma'am, of course. Then comes the next question. I have by Angarika Balakrishnan. I've read that some people are genetically predisposed to stress. Is this stress adaptive or maladaptive in nature? The first question added to it, and the second one is, can you pinpoint at which time point in life will this stress manifest?
Gosh, they're difficult questions, and I trust Angarika to ask me these tough questions. Hi, Angarika. Okay, so are people genetically predisposed to stress? Each of us carries a genetic bias, which will mean that our stress response pathways will be primed based on certain degree of genetic contribution. But this particular axis is highly pliable and responsive to environment. So in the case of this axis, I would say that it's the combination, of course, of your genes, but a huge component from your environment as well. So I would not say that you can be really, truly genetically predisposed to stress. You may be likely to have a pathway that is hyper reactive, but environment can override in many cases. So a very positive environment may actually buffer strongly against even some amount of a genetic predisposition. So then I would say that is not necessarily true that you're truly genetically predisposed. That there could be some degree of genetic predisposition, but environment's a bigger player. Can you pinpoint when this will show up? No, I wish we could. No. It manifests at multiple stages. So early stress can manifest with severity in adolescence, in adulthood, in late life. From all our rodent studies, we know that the most severe changes we see are actually once these animals start aging. That's when it's so dramatically obvious. In the early window, the system, even in adulthood, the system is trying to adapt and buffer and buffer and buffer. But you pay a cost for that kind of buffering. When you're trying to buffer so much, it catches up later. So yeah, unfortunately, I wish I could give you a cleaner answer for that. We don't really know.
Okay, ma'am. The other question is, would there also be a difference between cognitive development of single child compared to a child with multiple siblings?
I mean, it's a different environment for sure. I would not, I mean, even with time, nuclear households have become smaller and smaller. I mean, if you grew up in a household with grandparents and aunts and uncles, that's going to be different from a nuclear household. So of course, there's a difference in the environment. I would say that there is more likely to be greater difference between children brought up in an environment where there is stress versus no stress, as compared to whether they have a sibling or not. The sibling, I think, is less of a factor than the quality of the environment. So children being raised in an environment where there is high nurture, high care, high likelihood of being exposed to reading and creative development are obviously growing up in a much more nurturing environment. Siblings there would be good, not there also okay. I don't think there is that dramatic an effect. The dramatic effect happens in terms of the quality of care. So the siblings may be less of a factor than the nature of the environment.
Okay, ma'am. The other question is, does severe anxiety cause permanent damage?
Pathological clinical anxiety that is associated with a psychiatric disorder, not the kind of I'm feeling a little anxious kind of anxiety. Everybody feels slightly anxious. Not that kind of anxiety. The kind of anxiety that is almost phobic is associated with generalized anxiety disorder. Yeah, there are structural changes in the brain. I don't know if I would call anything permanent. I hope you will leave this entire lecture with the awareness that very rarely in the brain is anything so severe and so permanent. There is room for change. There's always room for repair. If that was the case and everything was permanent, then there's nothing that we can solve. Yes, there are certain neurological disorders where we can reduce the symptoms. We're not able to cure the disease. But even there is room for reducing the symptoms. So I would not want to go with the view that it is so impossible to repair. Yes, there will be structural changes in specific circuits of the brain, but there's profound effect of anxiolytic drugs. There is huge effect of cognitive behavior therapy, and many things like exercise, environmental change themselves bring around changes in the brain. So I would say that there's room for so much plasticity in the nervous system that it's almost better to focus on that than to view that things cannot be changed.
Okay, ma'am. So the other question is, ma'am, have you looked at impact of stress experienced at different age levels in subsequent life?
Yes, we have. So if you look at stress, I just showed you data from stress in the early window. Stress in the early window, the effects tend to be very, very long lasting. Stress in the juvenile window, not from our work, but from other groups, has shown that there is if the stress is relatively mild, it can actually prep the system for strong adaptive changes. Severe stress in the juvenile or adolescent window can also have very long lasting severe effects. In adulthood, more often than not, the effects of stress are short lived and you recover quite quickly from the effects of stress, except if the stress gets too severe. Then the effects can be quite bad. But in general, the answer is that the earlier and the more severe the stress, the worse and the longer lasting the effects. Yeah, so you buffer stress better as an adult than you do when you're a child. And that should tell us what we should not be doing to kids in schools and colleges. That window matters. That window really matters.
Yes, ma'am.
Especially in childhood.
Hello, ma'am. Can we move on to the next
Yeah.
question?
Sure.
Yes, the other question asked by Justin, do you think that there could be any correlation between early life stress and neurodegenerative disorders in humans? If yes, could you give some specific example?
Yes, there could be, because one of the phenotypes associated with early life stress is an accelerated or a worsening of aging phenotypes. And aging is, in a sense, a degenerative disorder where over a period of time, tissue degenerates, including the brain. And so there is a link there. People have actually used early life stress as a model for some of the worsening of neurodegenerative diseases. So a history of early trauma of that kind is considered to be one of the risk factors. It's not the only one, but it is definitely one of the risk factors. Yes. Alzheimer's disease being one example.
Okay, specific example. Let's move on
It's
to the
a
other
risk
question.
factor. It's not causative. It's just a risk factor. Sorry.
Risk factor. Okay, ma'am. The next one is in humans, in the particular case of deprivation of maternal care or paternal care, can the stress response be predicted based on the precedent cases? Are there common points?
That's a difficult one. I'm not sure exactly what the question is. So, in particular case of deprivation, maternal or paternal, can the stress response be predicted based on precedent cases, based on what's happened to other people who have gone through similar stress? To some extent, I mean, all that we conclude when we're generalizing based on what we've observed, we are saying that because people went through these kinds of stresses, this is what happens. We are making a generalized statement that may be applicable when you go through a similar stress, right? So, I mean, from what is known from clinical work, it is known that early childhood adversity or adverse childhood experience is a big risk factor for psychiatric disorders, for substance abuse, for alcohol abuse, for other health conditions. So that's been studied quite extensively. And it's not just maternal and paternal care. It can be, for example, I don't want you to walk away with these being the only caregivers. There's a lot of data that indicates that, for example, emotional abuse, sexual abuse, physical abuse in that early window is very strongly associated with enhanced risk for the eventual development of psychopathology. But a lot of this can be overridden. So there's clear data clinically as well as preclinically that foster care, adoption, sometimes individuals just need one person who believes in them. You have these incredible stories of a abusive environment, and then there's some teacher who comes along and believes in the child and believes in them thoroughly and just supports them thoroughly, and that child overrides very severe circumstances and shines and flowers and does incredibly well. So one should not go with the view that, yes, from the public health point of view, we want to reduce stress in early life for children and adolescents. Absolutely, because there's no question, the data is very clear. High stress in that early first decade is not good for a later life trajectory. It is a risk factor for lots of diseases. Okay. But it's not just maternal and paternal care, it is societal care in many ways. Our species, like for example, in monkeys, you can't do maternal separation as an experiment, it doesn't work. You need there are many other caregivers, and you will, even if you've seen monkeys, you will notice this. There will be an aunt, an uncle, a grandmother, somebody. So much the same way, primate societies are complex societies. Most of us will have had more than one caregiver. It's our mother, our father, grandparents, aunts, uncles, teachers, neighbors, friends. All of these, it literally takes a village to raise a child, is absolutely true. It really takes a village.
Okay, ma'am. We have another question. What does this buffering to handling stress particularly means physiologically?
Okay, physiologically, buffering means that you switch off the HPA axis. So when you buffer a stress response, so let's say you are under stress, okay? Let's give you a very simple example. You receive a let's imagine cold stress. Okay, you've gone into a room, it's very, very cold. It's extremely cold. So initially, because it's at say like 3 degrees Celsius or something, you are freezing. So your body is responding to a cold stress. So there's a cold stress shock. So initially, your hypothalamus will get activated. You will secrete cortisol. And cortisol will attempt to buffer. There will be an attempt to bring your thermostatic balance back to being able to handle that kind of cold. You cannot live at 3 degrees for long periods of time, but at least in the short term, your system is trying to handle that. And it's adapting to that shock of temperature. Much the same way for very high heat. You go into a very hot area, you're feeling extremely hot, then slowly the system tries to buffer and handle it. Buffering is an attempt to adapt. Okay. Much the same way, when you secrete cortisol, your system is going to switch off that cortisol response also, and then you get adapted to it. Very good way to think about it is think about the Mumbai local train. Any of you who have traveled by Mumbai local trains, when they are fully packed, will know that the first time you ever travel by train, it's a very severely stressful response. You cannot handle it. You may not even be able to enter the train because it's so crowded. You're not used to it. You have never traveled. You look at it and you say, this is impossible. I will never be able to even get into the train. 1 month later, you're traveling in the train and talking to people and listening to music and not even thinking about it. So now it's no longer stressful to you. Your system has buffered and handled the stress. You go every day by train. You don't even think, oh my God, how stressful. It's just become background noise. Your system has buffered and handled it. Most of the time, that is the case. You start a very difficult subject. For the first month, you're traumatized by the subject. You think, I'm never going to be able to do this. I will fail every exam. You keep studying. 1 month later, you're saying, what was I worrying about? It was fine. There was nothing wrong. I could handle it. Right? So it's not like every new event, when it's absolutely new, if there are a lot of challenges, initially it feels stressful, then you adapt.
Okay, ma'am. So next question. Does continuous exposure to stress cause damage to our brain cells?
Yes, it does, but depends on the kind of stress. If it's a short-lived stress and you switch it off quickly, then no. But if you keep being exposed to a stress for months and months and months and years, and you're not able to handle the stress, then yes, it is going to damage your hippocampus. Yes, no question about it. So, the question becomes what kind of, for example, going to war. That's a very severe stress, and our war veterans who have lived through wars or who have been exposed to those kind of conditions, are under a lot of stress, but many of them do adapt and learn how to handle it. And some of them undergo very severe experiences and develop post-traumatic stress disorder. But yeah, there are stressors that we are not expected to be exposed to, and not for long periods of time.
Um, Coming to the next question, for a person with trauma experienced early in life, what's your take on administrating preventive therapeutics in order to combat the disorder that set in later on in the life of the adult?
I would say if your therapeutics includes changing the environment, then that's the best kind of therapeutics. I would not want to say that we can prescriptively go in with a drug, imagining that that person is going to develop a disorder, because it's not a complete, it's a risk factor. It's not causative in its entirety. So early adversity and early stress is a higher risk for, so if you've had early stress, you're at higher risk of developing depression, anxiety. It's not a guarantee you're going to develop depression, anxiety at all. So you don't want to start treating in the possibility that you may get a disease. If we start doing that, we will be done for in many cases, because we cannot take drugs for everything that we may eventually develop. But I agree with you that you want to come in with a way to prevent it. And there are many things that could work. Quality of environment. Exercise is a big modulator of the brain, huge modulator. So coming in with positive interventions of that nature would perhaps help buffer. And they, in fact, exercise at the milder end of anxiety and depression is almost as effective as drugs. So, the benefit of this is something that people should think about, like in curriculum, when we're planning school curriculum or when you're planning college curriculum, leave room for sport. Maybe not competitive, horrible sport, but fun team sports. There is room for that. There is really a deep value to incorporating exercise as part of curriculum, especially and really incorporating it. What kids will do with that will probably have greater impact on their brain than sitting for hours and hours. Now, they're sitting for hours and hours in front of a Zoom screen listening to lectures. It is very important to modify our environment to incorporate these things. So I will agree with you on administering therapeutics of that nature, but not on agreeing as far as drugs. That I would worry about.
Okay, ma'am. The next question asked by Dr. Manju is that how can we manage stress in children? Does stress affect their overall development?
Yes, it can. So the most dramatic example of this is actually a really unfortunate example. In Romania during the post-Second World War, Romania, I mean, obviously Romania was a communist country then, and in Ceausescu's regime, there were a lot of orphanages set up where food was provided, clothing, food, shelter was provided, but there was no nurture, there was no warmth, there was no touch, there was no contact. And these children, with that, even though they were fed, they were nutritionally not deprived, ended up with a severe stunting and something called growth, basically dwarfism, stress dwarfism. So the most severe form of this is that when children are deprived of touch and contact and nurture, it can manifest to the extent that they become dwarfs. And they're not genetically dwarfs. They are developing dwarfism because the growth hormone pathway gets suppressed very severely under stress. And so, yes, even the height potential of the child can get affected. So there's no question that under that kind of severity, so there's a massive value. I mean, self-esteem, self-confidence, a sense of belief in oneself are built in that first decade. And, parents, teachers, grandparents, aunts, uncles impart this to children. So it is a collective responsibility. How does one manage stress in children? I guess the best way to manage it is we can remove it or reduce it. Or if we can't completely get rid of it, at least we can buffer it as best as we can by providing positive environment, positive reinforcers, etc. I mean, for example, a great example is how many children become totally demotivated to learn because of exams and because of exam performance anxiety. An exam performance is not an indicator of lifelong success or lifelong happiness. And yet it becomes a huge factor to deprive children of their joy of learning. And so if, as academics, we focus on the fact that the child is learning and not with the child's performance, focus on the child's learning and remove the performance association with it, we will save a lot of the future generation from unnecessarily being traumatized with the way schooling is done. I mean, I think there's a lot here to learn collectively as a society about what is happening to the brain in that window, and what we should be focusing on and what we should not be doing.
Yes, ma'am. Can we go for the other question? Yeah. Yes. Does what the mommy rat does in grooming, etc., when the baby rats are reunited, influence the cognitive performance?
Yes, it does. So, what the mother rat does is she licks and she grooms and she lifts them up and she moves them into the nest, and then she sits on top of them. And there's a lot of physical nurture. And the quality of that nurture has profound effects on how cognitive circuits develop. And so, work from the McGill lab, mini lab at McGill has really beautifully shown that cognitive development is also directly associated with quality of nurture. So the higher the quality of nurture, the greater the cognitive performance, and that's not very surprising. And that makes you start thinking about socioeconomic disparity, especially for a country like ours, and deprivation, and how across generations that can transmit because of the circumstances individuals live in. Poverty is a huge source of stress, massive source of stress, and it fragments family life by definition. So yeah, it's something to very deeply think about. It's a serious source of stress. Socioeconomic stability, where we don't have such steep disparity, and as a society we don't build the have not and have culture to this extreme extent, um, is something to worth considering collectively as a society. Because then when we see the consequences of, complain about the consequences are strife, eventually violence, fragmenting of society, and this emerges in that way. And then we question the emergence, but we're not seeing the roots. The roots are this disparity, poverty, extremes of wealth, and extremes of deprivation. So yeah, I mean, there are many fundamental questions to ask when one starts thinking about it, but absolutely cognitive performance is. And so sometimes, when we say things like, so many times we say, let it be a merit based society. What is merit? Are you counting the fact that for the last hundreds and hundreds of years, your family has had access to wealth, has had access to knowledge, has had access? That is not merit, that is privilege. So we carry privilege with us and we call it merit. Right? So when there is an attempt to balance out that privilege by trying to create equity and people protest against it, it is a conceptual. Think about when you say, oh, I did so well in something, you are thinking that it's all because of you. Well, it's also because you were given huge advantages. You were given advantages of wealth, nurture, access, all of this privilege that you carry, and some other child may have not had any of that. And then you're asking, even a small opportunity being provided to make it a little bit more equitable. We as a society of the privileged are protesting against equity because we view it as, oh, it's not giving merit a chance. Well, merit is a notion that is manufactured. Merit is based on privilege. And so I really feel that it opens up many, many fundamental questions, and neuroscience can inform some of this, because poverty is a massive early life stress.
Yes, ma'am. There are a lot of questions is being asked. Okay, we will move on. Are there any solutions or responses to long-time chronic stress at all? If not, many, then is there anything that can be done to prevent the state from worsening other than the medications?
Yeah. So, I mean, obviously, the best solution is to stop the stress. That would be the ideal, but that's not very, in many cases, that's not possible, right? So all you can do is reduce it or reduce the impact of it. That's what many times ends up happening. And so I would say, for example, there are things that one can do at the level of society, which involve access to healthcare, access to food, access to clothing, and access to education. These are rights that are fundamental human rights of any young child. And as a society, collectively, we are responsible for providing that. So that's something that's a societal responsibility. At the level of, and each of us can play a role at multiple levels. We can contribute to the societal contribution. There is our own individual family, there is our neighborhood, there is things that we see around us that we can buffer, and we can help in small ways. And sometimes one doesn't know what small help is going to have what impact when. So people just sometimes look at the scale of the problem and give up, because the problem is so large that it's impossible to fix in its entirety. But nothing is ever done that way. It's all in the small steps that one does, right? So for example, let's say if you have access in a school system, if you're teachers, you can change things for your classroom. You can have an impact at that level. And many of you will be college teachers, and when you're looking at it, and let's say somebody comes into your classroom, you don't know their life history. You're only evaluating based on what they're doing in your classroom. You don't know what history they carry with them. Just the being aware and open to the idea that people walking into your classroom carry a history with them, and maybe that you don't know the circumstances that they before they entered classroom, changes the manner in which you handle them. One thing I can tell you, believing in a person's ability is phenomenal, and it can have profound effects on what that person can go on to do. So sometimes just somebody having faith in a child or a young adult to succeed and do well can change their entire trajectory. And that's why there's such an important role for mentors, such an important role for role models, such an important role for someone who's a sponsor and believes in you. Such an important role teachers play. Undergraduate teachers, school teachers, is a profoundly important role. With what you do right, you can impact a person's life, many people's life. So I think that especially the education community and the academic community sometimes forgets the power they carry with them, and they can really impact many people's lives if they just choose to do so.
Yes, ma'am, sure. The next one is, can too much comfort in early life of mouse cause negative effects on their brain? I mean, the person means that can complete lack of stress in a mouse life will prevent the ability of the mouse to develop stress responses.
Interesting. I don't know if that's even possible. I mean, I don't know what too much comfort is, and in the animal house, they're already quite comfortable because they're being provided food. And so in the laboratory situation, you're already providing so many things that you're looking at a more cushy existence for sure. The question I think you're saying is, if you have zero challenges, will you be able to handle a challenge when you see one? Right? I mean, let me rephrase that. I'm not sure there exists a existence which has zero challenges, but I would agree with you that children or adults taking responsibility is critical to their learning to take responsibility later on. And so certainly if you raise children with absolutely zero sense of responsibility to themselves, their environment, and their surroundings, they will grow up to be adults who take zero interest in any of that. And so to that extent, I think it matters hugely that what individuals do in terms of the responsibilities given to children. I think that responsibilities are not stress. Responsibilities are a privilege. I mean, with responsibility, there is freedom comes with responsibility, but you do have to give responsibilities to children as well. And they are more than capable of executing those responsibilities. When given appropriate responsibilities, they will execute. So I'm just flipping that into saying that I think children should be given responsibilities from that perspective. And certainly a cushy existence to the point where the person has to do absolutely nothing is not a viable way. We all learn by learning things. So, you share responsibilities of your family as a child. For example, a great example is during this entire pandemic. If you don't do anything and you watch your parents clean, cook, handle things, and you don't contribute in any way, that's not a great way to develop self-esteem. Whereas if you contribute around the house and become a contributing member, that contributes to your feeling like you're also contributing to whatever the entire family unit is doing. So I think zero stress is a little unlikely, but some responsibilities are very good.
Yes, sure, ma'am. We have a lot of questions in the chat box. I think it's a time up, and let's conclude the session. And the remaining questions will be, ma'am will answer through email. So don't participants don't be disappointed. I'm sure will answer those questions. So now it's a time for lunch break. Thank you, ma'am. Thank you so much for the amazing session and for answering all those questions beautifully. And it was a very informative session, ma'am. So may we conclude the session, ma'am? Okay. Outcomes. The forenoon session has come to an end. So participants, please join through the third webinar link given in the chat box of WhatsApp. So hope all will join at 2 p.m. Sharp 2 p.m. Thank you all. Thank you.
1 minute, I will share the feedback link right now.
Okay.
So that they can do it. Okay.
Okay. The feedback link will be available on the chat box. Participants, please be sure to fill them before leaving. A gentle reminder to all the participants, e-certificates will be provided only those who attend the 2-day sessions. Thank you.
Feedback form is available in the chat box. Kindly all participants fill that and make sure you get the certificate after the 2-day workshop.